This slideshow will help explain the clinical test known as whole exome sequencing, abbreviated WES. Whole exome sequencing is used to determine if a genetic basis exists for a given patient's condition. In order to better understand this clinical test, it is important to first review some key concepts. The human body is comprised of cells. Most of these cells contain DNA, which is often concentrated in a region of the cell known as the nucleus. The DNA molecule can be organized into structures called chromosomes and stores information using the 4 bases A, T, C, and G. Genes are defined segments of DNA which tell our bodies how to work. More precisely, genes can code for specific proteins. These proteins then direct the structure and function of our bodies. Genes themselves can be split into different parts. These parts are known as exons and introns. The exon is the portion of the gene believed to be most responsible for telling our bodies how to work. Introns are intervening sequences that are removed prior to the assembly of a protein. The exons present in your genetic material are known collectively as the exome. Imagine you are studying from a textbook. If you knew what questions were going to be asked on your exam, you could focus on the most important pieces of information given by that text. Whole exome sequencing works in much the same way, by allowing us to focus on the exons, which are the most important parts of a gene. Roughly 85% of all known disease-causing variants are found within the exome. This makes whole exome sequencing an extremely comprehensive and cost-effective test. We all have genetic differences which distinguish us from one another and add to the overall diversity of the population. Genetic variations can be classified based upon their potential to cause disease. Benign polymorphisms are natural variations in a gene that are not known to have adverse health effects. Variants of uncertain significance, abbreviated VUS, are more recently discovered changes in our genetic material, and their effects have not yet been determined. Mutations are genetic variations that cause disease or are known to be associated with an increased risk for health problems. In order to undergo this clinical test, blood is drawn from the patient. It is also preferred that blood is drawn from both biological parents. Samples collected from the parents of the individual undergoing testing help to interpret the results of whole exome sequencing. Blood collected from the parents allows the lab to examine variations shared between the parents and their child, thus helping to determine if these changes adversely affect health. There are limitations to whole exome sequencing. Whole exome sequencing only examines about 90 to 95% of the exome. Therefore, a mutation in the part of the exome that is not tested would not be identified. For this reason, a negative test result does not necessarily rule out a hereditary cause. It is likely that new research could change the interpretation of your results, so additional testing should be considered in the future. You should also be aware that non-paternity or adoption status could be identified by this clinical test. Since benign variations are not associated with adverse health effects, they will likely not be reported in your test results. Variants of uncertain significance are not currently associated with adverse health risks. Thus, health management would only change as new research regarding these variants becomes available. Mutations are associated with an increased risk to develop health problems or may be known to cause certain conditions. Identifying these mutations helps healthcare providers determine how best to clinically manage these conditions and reduce the risk for developing problems in the future. Family members of a patient with an identified mutation could also be at a higher risk for carrying the same mutation. Therefore, your test results could also be helpful in informing your family's healthcare decisions. Whole exome sequencing offers different types of positive results. It is possible that you may not want to be informed of the results in all cases. Since roughly 20,000 genes are examined, there is a possibility that mutations may be identified that are unrelated to your known health conditions. These are referred to as incidental findings and have the potential to alter your clinical management. One example could be the identification of a mutation which is associated with an increased risk for developing cancer. It is important to be aware that you are able to opt out of receiving these results if you choose to do so. You also have the choice to opt out of receiving positive results concerning carrier status or pharmacogenetics. Carrier status means that you possess a mutation that does not directly affect your health, but could indicate an increased risk for any children you may have. Pharmacogenetic changes are related to the way in which your body uses or metabolizes drugs. Identification of pharmacogenetic changes could possibly be used to help determine which prescriptions will work best for you. A blood draw will be conducted today. It takes approximately 4 to 6 months to receive the results of the test. Informed consent should be given regarding the implications of genetic testing, the limitations of whole exome sequencing, and the issue of non-paternity. Insurance information should also be obtained, and any out-of-pocket costs should have been previously discussed with you. We hope this presentation has improved your understanding of whole exome sequencing. Please be sure to schedule an appointment to discuss your results with a genetic counselor.